Thank you very much, Kelvin. I'm, as usual, very pleased to be here in this club. And I am going to discuss today some of the studies we've done with centenarians. How did I finally decide to work on centenarians? Some of you may remember we've been involved in work on longevity genes. So we were essentially putting genes into animals and seeing how they increase the longevity. And as far as I am concerned, we did work on P53, P16, telomerase, and Ras GRF1. So I thought, why not to see how these and other genes are in humans, in centenarians. Centenarians are unique in that there's no experimental animal for that. Imagine that I went to my student and said, grow 5,000 mice and we use the last one. So that's a centenarian, 1 in 5,000. So that's a unique group. But also personally, this was my grandmother and she was there celebrating her 100th birthday. So I hope I have the gene, but I'll give you another glimpse. I will discuss, there will be a prologue, and then centenarians regulate microRNA, the biogenesis, then mRNAs controlled by microRNAs, then the newest experiments, apart from this part, all is unpublished. The prologue is that centenarians not only live a lot, but they live very well. Their health span approaches their lifespan. And I have anecdotal evidence for this. This is my mother, and this is my daughter celebrating her 24th birthday. A couple of months ago, she phoned the grandma and said, "Grandma, come tomorrow because there's a party." And I was there, exhausted, well past midnight, and they were dancing. And I was hoping that I could walk with my mother to her place where she lives alone. So I think that she's an example. I hope that the gene passes on, at least, if not to her, but at least to the daughter. And so they not only have a very prolonged longevity, but they're very good health span. And that's why I'm trying to learn from them so that eventually we can apply this knowledge to the general population. The first thing I want to show you, and this, I hope I convey a message that is like a flow. It's not different points of different studies, but a continuum in the last 2-3 years in the lab. These persons upregulate the microRNA. The strategies that we have blood from a centenarian cohort, and then we have octogenarians and young of the same area near Valencia, and we are performing a microRNA analysis, and then we want to see which mRNAs are controlled by these microRNAs, and then we will do a subnetwork analysis, and I will explain all this. In my 20 years of studies in aging, the work of Paula Timiras, a very remarkable woman that made the point that all persons lose capacity to control. The basal glycemia doesn't change, it's the glucose tolerance test that changes. So it's essentially, they lose capacity to control. So I thought that instead of studying mutations and all that, I would study these then relatively new molecules, microRNAs, that were able to control transcription. All right, the first thing we found, we did the microRNome, and the first thing we were surprised to find is that young and octogenarians are very different, but young and centenarians are very similar in terms of microRNA expression. So these centenarians are really like young. So when we compared the microRNAs that were upregulated, microRNAs from centenarians beat young 7-0. Octogenarians lose to young 1-50. Centenarians beat octogenarians by far 102-1. So there's a vast defeat for octogenarians when compared with the centenarians. There's only this gene that is upregulated in both, and this is relevant. So of the 7, that one gene we haven't taken, and there are 6 others that are these ones, of which 3, and this is carna K comes from Cajal, our neuroscientist, so who had a knack to find important things, and here comes again. So we have studied these three ones that, whose function we know, plus the SCARNA. And these are the functions. I just brought it because I know we've been filming, and maybe that you want to have a look at each one. And then SCARNA, who is located in Cajal's bodies and controls RNA polymerase II, and this will come into the picture later on. So we did the PCR. When you do the microarrays, you always want to do the PCR of the relevant genes, and we found that yes, centenarians upregulate SCARNA17 and the 3 mirs. So we had these 4. And the message that centenarians upregulate these regulatory genes, and that's the only part that was published later last year. So how come, so if you want to have details there, how come that centenarians upregulate miR? Why? So then we studied, and we are studying, the microRNA biogenesis. And microRNA biogenesis, this is the second arrow, is this. So RNA polymerase II, sorry, synthesizes pre-microRNA, which is broken down by drosha to form pre-microRNA, and this is exported by exportin, and dicer makes the final one, the final mature double-stranded RNA. All right, so we thought that this might be important to show how come that these centenarians, have this capacity to upregulate microRNAs. And this one came to my mind only last month. And then this paper in Nature appeared, deregulation of microRNA has been linked to diverse pathological processes, including cancer. And microRNAs are involved in the cellular response to stress. So the thing is there. And one of the references in this paper is this older one, modulation of microRNA processing by p53, and I, with Maria Velasco and Manuel Serrano, were involved in finding that this was one of the longevity genes I was mentioning to you, ARF, p16, and p53, and as I was saying, you put the gene in the animal and the animal lives longer, all right? And this was, our job was to show that p53 behaves as an antioxidant. All right, so we did the processing of microRNAs and found that this comes from the microchips, the macroarrays. But I'll discuss in more detail the results when we did the PCR analysis of the relevant genes. And we found that drosha and dicer are completely down-regulated in octogenarians. In 25 years working in aging, I have never seen an enzyme that is blocked like dicer. So, not the centenarians. This does not explain that centenarians have more level. Centenarians have more or less the same, but it's very significant, the lowering in octogenarians in agreement with the results obtained by microarrays. But exporting works the other way around. Centenarians have higher levels than young, and let alone octogenarians, very significant. So we think that the pattern of expression of microRNAs in centenarians and in normal aging, i.e. octogenarians, can be explained because octogenarians have very low drosha, which is normal in centenarians, and very low dicer, which is normal in octogenarians. But so centenarians having normal levels of dicer and drosha have a higher level than anyone in exporting. So the analysis of the pathway of the maturation of if you will, biogenesis of microRNA biology explains why these special people have a special maturation of microRNAs. And this is very new from the lab now, but we hope that with more N, we will be able to tune better. But the idea is drosha and dicer are down-regulated in normal aging, but not in centenarians. And then exporting is upregulated in centenarians. So having had a look at the microRNA biogenesis, we then said, all right, let us see how mRNA is controlled in centenarians. And we had a hypothesis, and it's the following. So what we did, and the third arrow, is then studying the mRNA, this, and with this, and this, we were expecting something like this. So centenarians and octogenarians are very different. The data are not as clear as with microRNA, but still centenarians, the mRNA of centenarians and octogenarians is very different. And if you compare, the figures are much more than with microRNAs. There's 12,000 difference between centenarians versus old. So centenarians are really different from old. And these figures comes when you compare centenarians versus young and octogenarians versus young. And if you do the Venn diagram, you come up with 1,700 genes that are different in centenarians from octogenarians and from young persons. Of those, the hypothesis is, let us see which are the ones that are controlled by the microRNA that we had found that to be different in centenarians. So of those, we wanted to combine which of these genes were controlled by these microRNAs, and we came up with 51 genes. And then we did a sub-network analysis. And then when Eva Serna came to me and said, we're going to perform a sub-network analysis, I said, I put my foot down and you tell me what is a sub-network. And sub-network is a group of genes connected structurally or functionally to one gene. So what you're going to see is a rosette of genes converging on one. That's a sub-network. So when I understood it, I said, all right, do the analysis. So, and I'm going to show you what happens, for instance, when you combine the 51 genes controlled by the 3 microRNAs specific for centenarians, all right? And this is what you find. T-cell receptor, when you compare centenarians versus young, blue is higher, red is lower. The significant thing is that if you compare octogenarian with young, it's almost a mirror image. So much so that I thought that it might be that I was doing some circular way of thinking. So I went to the mathematicians in Valencia and they said, no. So I went to my friends in Karolinska in Stockholm, Jesper Tegner, the head of the department in biostatistics, and they've been looking at the data and saying, no, it's not a mathematical error, it's simply that this is red. Okay, there you have one red and red, but red, blue, blue, red. So what is upregulated in centenarians tends to be down. There are some common, so that's where it's not a mathematical error. Tends to be down-regulated in octogenarians. Okay? And what we've found, this sub-network converging in special genes, and there are 6 of them. T-cell receptor, interferon gamma, SP1, a transcription factor, always the mirror image, right? TGF-beta1, tumor growth factor, and TNF, tumor necrosis factor. These are the 6 genes that we have been able to pinpoint when studying all this group of genes. And therefore, if you put them together, you find this, simply all in one slide. You won't be surprised that if you compare centenarians versus young, when you compare octogenarians versus young, is the opposite image. Okay? So these genes, if you do a second derivative and say, are these related to something? Are these genes that we have found related to something? And they are related to 4 genes. BCL-XL, FAS ligand, and CCL5. Of those, 3 have to do with apoptosis. FAS and FAS ligand activate the extrinsic pathway of apoptosis. BCL-XL inhibits the intrinsic mitochondrial pathway of apoptosis. And all these tend to be controlled or related to BCL-XL. Then, of course, we went and tested BCL-XL by PCR, and yes, it's upregulated in centenarians and not in octogenarians. So we believe that the BCL2 family, and especially BCL-XL, is very important in conveying a special function for the centenarians. Now, my question now, what we're doing now, is do we have functional? Can we show functional things? And remember, cells from centenarians, and we need fresh cells. So I'm limited by the amount of sample. I cannot go and bleed some centenarian because the daughter, normally taking care of the mom, will say, no, you bleed my mother once. You're not coming to bleed my mother once a week. So we limited with the samples, but yet I think shortly I'll be able, from other cohorts, to show you data on how these centenarians have a fantastic immune function. So far I can tell you, and this is the last little line, that yes, this is a mitochondrial protein, and so we're back to square one. You may not remember, but my first serious paper on aging was the thesis of Juan Sastre, who spoke this morning when he was working with me, and we proved, and it was in hepatology already 20 years ago, published, that mitochondria are damaged in aging. They were proposed, there were proposals that were damaged. Bruce Ames came out with a very similar data a few months later, so we agreed on that. So, I was pleased to see that this is a mitochondrial protein, and with some antioxidant properties, and if you look at the MDL levels in plasma, they're slightly lower, significantly lower in centenarians, and protein oxidations lower in centenarians than in young. And if you look at BCL-XL, inhibits BAX, prevents cytochrome C release, and this I was not aware, cytochrome C is shed. It's not that the cells break, it's shed to plasma. So one can measure and expect to find less cytochrome C in centenarians, even though in plasma you don't expect big changes, but there could be. So we measured cytochrome C in plasma of the centenarians. First of all, we measured in cells how the mitochondrial membrane potential is very much higher in centenarians than in young or old persons, and this was done by flow cytometry. And then we checked cytochrome C levels in plasma, and sure enough, they're lower. So apparently, BCL-XL is having a very significant effect in terms of function. When you study cells we study mononuclear cells because that's the only cells we can have an access to. There's no way that they're going to give us a muscle biopsy, and I would not want to do that. So we're limited to what we can look at, and these are, and remember, these are blood cells. Remember, no animal model for centenarians, because you cannot, even with flies, to have the last fly of 5,000. So, BCL-XL, blocking BAX, preventing the intrinsic pathway of apoptosis, and I'm now trying to get cells to see the extrinsic pathway, the fast ligand mediated, but my view is that centenarians have a low intrinsic pathway. When the cells are more or less all right, they leave them, but if you challenge the cell and you give a genotoxic, they kill the cell immediately. So the external pathway of apoptosis is activated, the intrinsic is sort of blocked by BCL-XL. And the epilogue, the conclusions. I hope I conveyed the idea of a flow, of how this has more or less gone and not as kind of independent studies. So the conclusion or epilogue is centenarians exhibit extreme longevity and a remarkable compression of morbidity. That's a fact that we see. Their health span approximates their lifespan, and my mother is an example. They have an outstanding capacity to maintain homeostatic mechanisms, so Paola Timira was in the end right. And this turns out when we study genes, they upregulate microRNAs. They have a special biosynthesis or maturation of these microRNAs. And after studying the mRNAs and the microRNAs, we think that BCL-XL gene expression seems to be critical to convey, to give you the possibility of becoming a centenarian. I have to finish. Please have in mind that this is a very large team working with a large group of clinicians led by Juan Antonio Avellana near Valencia. This is a very nice nurse, Paula Sanchez, and she treats the centenarians with a lot of love, so they give them the blood. This is Eva Serna, who does all this microRNAs analysis, and this is my closer group, Juan Gambini, Vicente Bonet, who is sitting somewhere around there. This is Kira Mohamed. She's doing a lot of the field work. Chelo Borras, who's been working on aging. And I wanted to outline here the French contingent. They're not involved, but because they're here, they come from Arlette's lab, and they've been working for about a year or two in the lab, Frédéric Derbré and Thomas. And finally, I want that you realize that these people are all smiling. They make my life, professional life, very pleasant. So I thank them, and I thank you for your attention. Thank you very much.
So this paper is now open for questions, and I'd like to congratulate Pepe on doing a little bit of work since I last saw you.
Yes.
Quite amazing stuff. Thank you. Great job. Yeah. Do you need a microphone?
I'm searching for the microphone, by the way. So that's,
Great. Does it work? Yes.
Okay, it works. You mentioned that some of these microRNA control the expression of RNA. Are these genes validated target of this microRNA, or you just believe that they may be regulated on the basis of the sequence? And another little question is the cohort of octogenarians. Could it be a little, well, the percentage of putative centenarian is very low, but could the cohort of octogenarian be contaminated by some potential centenarian?
To answer the first question is the genes, the critical genes we validate by PCR, right? Okay, so and the sequence, yes, is not valid. Is based on the structure, and on this eventually on the subnetwork analysis, right? So it's functionally or structurally related, and yes, first, the cohort of octogenarians, and that I failed to say this, but my friends in geriatrics say they're not always octogenarians, the early octogenarians, so it's close to 78 to 82, all right? And yes, there'll be a contamination with centenarians, and that's what we're looking for. We're trying to find if we can spot the one. Next, we're trying to do is to find genetically enriched octogenarians. So octogenarians who are sons or daughters of centenarians, like my mother is. It's too late for my mother, and she won't give me blood. But I said, because she is already non-nonagenarian, but some 10 years ago I should have taken blood from my mother and from my grandmother, and then, but I wasn't studying centenarians then.
What's the percentage of octogenarians that reach centenarians?
Well, 1 in 3,000-4,000, because the majority of people reach the age of 80 these days.
Yeah. So,
but then they fall like...
So you'll have to be sampling 10,000 people to get just a couple.
We are sampling to 250,000.
250,000.
Not sampling. We have 250,000 area of persons that are phenotyped by the clinics in the hospital near Valencia, and there's 250,000. And for those we have found 31 octogenarians, centenarians, and then the mathematicians came to calculate the number of centenarian of octogenarians. So yes, 100, a quarter of a million person.
So just a very nice talk, but how do you separate cause and results? So by reaching 100, these genes may not be causal at all for reaching 100. They just may be the result of what's left when you age up to 100. The gene expression changes, but it may not be the reason that they made 100.
Agreed. The two approaches. Yes, I asked myself that question. How am I not studying proximity of death? Because the centenarians have an average life expectancy of 1 year. We have two approaches for that, but you're completely right. I mean, in purely logical terms, I don't think I can solve the question. But there are two approaches. One is the centenarians that are, octogenarians that are the sons of centenarians. That's one approach. So we're looking for families. And the second approach is measuring physiological activities of mononuclear cells and trying to see whether those cells are still doing better. And that's it, as far as I can go. But thank you for the question.
It would be very nice if we could get a case control study with a super centenarian. There are about 500 of them in the world. And if you can identify and get just one just to do these analyses with a single individual would be fantastic. But you're always thinking ahead.
If you help me find the super centenarians, we do all the analysis. Yes, there's a cohort in Leiden.
Quick question. Great talk. Just a quick question on the selection of the cohorts, especially the young. How old were they? You know, how many individuals did you have in each of the groups? People tend to believe that aging starts after 40, 45, depends on who you want to believe. So I think it's important, you know, cohort studies are very dangerous in the sense to tease out causality. So can you talk a little bit about the young group that you selected and how you really going to compare this? How important is that group?
That's very critical because they are the controls, and we asked ourselves that very question. And we were guided by Jaime Miquel, who early on got me really into gerontology, and he said that there's no age there's no age to become old. That you're old at age 15 if you want to do gymnastics, so artistic gymnastics. So we decided. He said otherwise 30 is the year when one should consider that aging starts. So our people were 30-year-old. There were a number of criteria of exclusion that we did not include in the centenarians, or you ran out of centenarians, and they were from the same area, and they were 30 plus minus 2. And the octogenarians, which are not octogenarians, 80 plus minus 2. And we made the point of having all from the same area, Christian, area of Alcira, and we're validating now with another cohort because we founded the Spanish Centenarian Study Group, so we have now contacts with 4 or 5 cohorts of centenarians. That's
Did you do any functional analysis?
Yes. Christian is asking me about the functional analysis. You mean they were very well phenotyped, very well in terms of frailty especially, and the cellular functional analysis is being done now by Mónica de la Fuente in Madrid. You know.
We have 2 more questions. There's one up here.
Yes. Did you compare the status of glutathione and NRF2 in the population?
I should have done, because my thesis was on glutathione. No. The question is this. To measure glutathione, we know from my own studies, you have to take the sample in a very special way. This is putting N-ethylmalamide, so you need 2 tubes, one for N-ethylmalamide and one for GSH. Otherwise, the ratio is meaningless. So I thought of that. Thank you for the question. But I decided not to study glutathione even if it is my molecule, because of the difficulty in obtaining the samples. And you want samples for DNA, RNA, microRNA, then the clinician wants samples for glucose, cholesterol, and all these kind of things. So I decided not to use glutathione, not to measure glutathione for that reason.
Congratulations, have good gene.
Where are you?
Me here.
Ah.
How about their muscle functions? That's an easy one to measure with the Centenarians. That would be.
I don't think the clinician followed Carmen's advice, because we wanted them to measure, I mean, obvious, grip strength and that kind of function. You cannot, the biopsy is out of the way, so biochemical measurements of function, but grip strength, all these things that we measure for frailty, they may have measured those. But I don't think in this first cohort. The cohort they're taking now, they are involved in that, and certainly in Toledo, Spain, the original Toledo, there's a cohort, and they are going to concentrate on those things in the context of frailty. So we will have news about this in a couple of years' time.
Okay, last question, Volker.
Thank you. You make me thinking. Maybe you already answered before, but back to focus. Definitely, from young to octogenarians, you have to change in the gene expression. Then you have a different, let's say, phenotype from 80 to 100 years. And this could be most likely, again, a change in gene expression. So in other words, there is just a different pattern of gene expression in different ages, and then these guys have that gene expression because they are centenary. And they are not centenary because they have this expression. Yeah, I was exactly the same. But I was focusing on the other side of the story. Because you have no doubt that from young to octogenarians, it's just a matter of gene expression. I'm just talking about similarity,
My conclusion, first of all, we are also sequencing the exome. And there are different genes, but for common mutations, you need huge numbers. Tom Pearls already had this problem, and they had to withdraw a paper from Science. So that's why. But yet we're sequencing the exome of families. Second, for mutations. For gene expression, my conclusion is that the regulation, if mRNA, as said generally, control gene expression, is the control of gene expression that is less tight in octogenarians than in young, and than in centenarians. That's good. And you could tell me in the end, like before, so you are studying survivors. Yes, but I want to know what do they have in case we can apply to ourselves and be here in 30 years' time.
Okay,
Thank you.
Thank Dr. Vina very much.